matter which tissue? So do different do the mitochondria in the different tissues produce? Are they different? I mean, do they work in different ways, and do they produce different peptides?
Absolutely. And I think it's not really appreciated even by many scientists that the mitochondria from, say, brain and liver and muscle are very, very different. They look different by electron microscopy. If you isolate mitochondria and do a proteomic analysis, you see very different distribution of proteomic results. If you look at the function, they're quite different in their energetic profile. So it's no surprise that we identify different profiles of expression of mitochondrial peptides. One of the techniques that we've recently developed is the mitochondrial equivalent of what's known as RNA-seq. RNA-seq is a technique that was developed to measure the RNA from tissue samples and look at all the 20,000 genes that express RNA and eventually proteins and see what goes up and what goes down in various conditions. The standard RNA-seq does not look at microproteins, certainly not in the mitochondria, and we've developed a method to specifically analyze the expression of mitochondrial peptide at the RNA level, and they're very different from tissue to tissue, and it's very sensitive to disease states and interventions of all sorts. And indeed, the profile of mitochondrial peptide that are produced by mitochondria from different tissue is very different.
Interesting. Yeah, because I always thought of mitochondria as being the same, like everywhere.
Kind of like standard issue, you stick them in cells and they just make energy, but it's not the case.
That's interesting. Yes.
There's a concept of mitochondrial transplant, right? So you can take mitochondria and put them into tissues, and taking them from one type of tissue to another can have profound effects. It's still a concept that is in development, and it's not quite ready for even clinical trials. But which mitochondria you start with is going to have a major effect on the results you're going to get.
So one question on mitochondria and aging. So does the DNA damage in a mitochondria just accumulate and accumulate like over our whole lifespan, or do they have I don't know, can they reset?
Right. So the mitochondria employ a very different model than normal mammalian cells. They are essentially continue to function the way unicellular bacterial-like organisms do. They divide much more. First of all, a cell has two sets of chromosomes and one cell, and the cell divides as needed in the tissue. But each cell has hundreds, potentially thousands of mitochondria. And the mitochondria divide in an asynchronous fashion with the rest of the cell. And there are many mitochondria, and they undergo a process known as mitophagy, which is kind of like autophagy, where a whole cell gets sort of eliminated when it's done with its role. Poorly functioning or sort of not quite 100% effective mitochondria gets eliminated through the concept of mitophagy. And even though we said that there's a lot of damage that happens to mitochondria as you age, a lot of these less than perfect mitochondria get eliminated through this mitophagy process. So you're always going to have some functioning mitochondria. But if you're, 18, your body is really good at getting rid of all the abnormal mitochondria and keeping like a really good cohort of fully functioning mitochondria. By the time you reach my age, there are more and more of these sort of semi-functional ones that are trying to do the job. By the time you get to 100, there's even less quality control there. And that happens at a steeper rate than some other components of the cell, all of which age, but mitochondria seem to age fastest among the different elements in the body.